Naomi, can you hear me? I can see one person there, but it's not showing up here in the live stream. But I can see your name here. So if you can hear me, you can give me a thumbs up. You can do that in the chat. Awesome. So you can tell me where you're from, what grade you're in. You can give your school a shout out as well. Ninth grade. Awesome. Well, let's go ahead and get started. I hope you're not nervous about the assessment. I hope you're excited about it. I hope you're confident. I'm just here to provide you a little bit of support, maybe answer some questions that you may have about some of the particular standards or maybe some of the particular units that you guys have covered already. So my name is Dr. Christopher Holmes. I am one of the co-founders of STEAM Connect Foundation, and we're just a nonprofit, and we're here just to provide some free support to you as it relates to the biology milestone assessments that is fast approaching. So how long do you have in your district or at your school before you guys take the test? One month. Okay, you got some good time. All right. So I'm going to start with just about 15 questions. And the best way for you, since you are live and you can use the chat, is to just throw your answer in the chat. These are going to be kind of our warm-up questions, right? These are kind of basic. These are things you should know before we get into some of the deeper questions. But before I do that, if you can look here on the screen, you'll see that the assessment is broken down into various components. You have 5 units that you guys have or will be covering this school year. So cells, genetics and heredity, classification, ecology, and the theory of evolution. You can see those standards listed here in the middle. And then you could see approximately how many questions you're going to have per unit, and then what that translates into as it relates to a percentage. So the heaviest area is going to be ecology, and that's going to be standard 5. The next is going to be cells. The next is going to be genetics and then cells. And so we'll hit those 3 units really heavy while we're together for this time. And then we'll throw in a couple of questions as it relates to evolution and a couple of questions as it relates to classification. Okay. Any questions you have for me before we get started? All right. I hope you have paper and pencil out, or you're typing on your computer. Take some notes. If there's something that we go over that you still don't understand, I'm going to throw my email address in at the end. You can always shoot me an email if you have more questions. We can get back together throughout the next month to kind of help you get prepared. All right, so let's look at just some sample questions. All right, and again, these are just review. And as you can see, I'm using Quizzes to do that. This is the GMAS review from Quizzes. All right, so for you, just throw the color in the chat that you think is the right answer. So let me reshare this because that's a whole new tab that you cannot see. All right, first question. Which do DNA and RNA have in common? You can go blue, green, yellow, or red in the chat. Or she says teal. All right. She's a fashion. She's a color girl. All right. Which do they have in common? The answer is correct. They both have ribo. Remember ribonucleic acid? Um, they both contain phosphate. To remember, if you think about the backbone of DNA, they're made of the alternating phosphates groups. Okay, so we know DNA is double-stranded. We know RNA contains uracil and not thymine. But whether it's double-stranded or single-stranded, which RNA is, both of those have or have to have phosphate groups. Okay, so they have a sugar, they have a phosphate, and then they have that nucleotide or that base right in the middle. The ribose specifically relates to RNA. So when I said the sugar, the phosphate, and then the nucleotide, RNA has the sugar ribose, while DNA has the sugar deoxyribose. So that's what makes them different, is the type of sugar that they have. So let's look at this really quickly as just an example. All right, so if we look here, let's make this, look to the image to the right, okay? So you see these circles and then these pentagon shapes? These represents your ribose and your sugars. Ribose, sugar, and then your phosphate group. So phosphate, and then you have your 5-carbon sugar. Phosphate, the circle, your 5-carbon sugar. Phosphate, 5-carbon sugar. And attached to the sugar, you have your bases, right? Every piece of genetic material, whether it's DNA or RNA, will all have phosphates, okay? But DNA has a particular type of sugar, which is deoxyribose, and RNA has the sugar ribose. And again, when you're looking at RNA, remember, it is single stranded. So you won't see the double strands like you see here in this double helix. That is only specific to DNA. All right, DNA is double stranded, RNA is single stranded, and it has the sugar ribose. Okay, or ribose. Does that make sense? Okay, awesome. Let's go back to the questions. All right, so now we're talking about just DNA. We know DNA is double stranded. So I want you to find the complementary RNA strand sequence to the DNA sequence ATTGCA. All right, the complementary RNA strand to the DNA sequence ATTGCA. Which color is it? And think about what we just said about what's different between DNA and RNA. All right, let's see what you said. She says yellow. Okay, so let's look at these. So we know that A in RNA will pair with U. So you got that part correct. So we can eliminate the blue and the red. So it's either green or yellow. Okay, so A pairs with U, T pairs with A because RNA still has adenine. So UAA, G pairs with C, G pairs with, C pairs with G, and then A again will pair with U. So that's correct. You will not have any T's or thymines in your RNA. All right, very good. Next question. Which defines a codon in DNA or messenger RNA? What is a codon? What is a codon? That is correct. It is 3 bases. So AAC will give or code for a particular amino acid. Okay. And we know those amino acids eventually all link together and they create proteins. All right. Very good. All right. Looking at this pair of sequences. If we look at the top one and we look at the bottom one, what type of mutation occurred? So first find how they're different and then tell me what type of mutation occurred. All right, so you will see that. Look at the fourth base in each one of those. All right, so you're correct in the sense that the G and the C are not the same, right? So TGA, they're all the same here. And then you have CCA, and then at the bottom you have GCA. All right, if you have a frame shift mutation, everything after the mutation will also change, okay? Which means this would likely be G something else, something else. It would not look very similar to the very top one after the mutation. Okay, same thing if you had an insertion, you would have G, and then following this, it would be CC, and if you went further, it would be A, which means you inserted a G, and then you would continue on with the rest of it. So TGA insert a G, then CCA. Well, you know it's not insertion because this says just CA. Okay, if you deleted something, then it would shift to the right. So you don't have any type of shift. You only have a change in the G and in the C. And when you only have a change in just a letter, then the answer is a what? Substitution. All right, so I substituted G for C, but it didn't cause that strand to shift either left or right. All right, both deletions and insertions are types of frame shift mutations. Okay, because I'm deleting something, it's going to shift to the left, or I'm inserting something, it's going to shift to the right, and nothing shifts. You just have a change in that one base. All right, let's look at some other questions. Let's kind of get out of DNA for a while. Let's look at this. All right, which is not a phase of the cell cycle. Which is not a phase of the cell cycle. You remember you have G1, you have S, you have G2, and you have M, right? And then you have the phase where the cell splits in half. So when you think about a cell going through cell division, right? Or just replication, let's say that. So a cell is getting ready to replicate. We know cell replication in its general form, we normally describe it as mitosis, right? Cells are replicating. That whole process is called mitosis. So the entire cycle is mitosis. G1, S, and G2 make up interphase before we get to the actual splitting of the cell. And then when the cell actually splits from 1 into 2, that process of splitting is called cytokinesis. But we never frame the cell cycle as duplication, okay? So the cell cycle does involve mitosis. It does involve cytokinesis. It also involves interphase, but it does not include what we call just simple duplication. Okay, let's look at that again. There's several parts to it. That's easily confused by a lot of students, right? Because it can be hard because there are several parts to it. So let's look at all the parts again. I want to visually show you the cell cycle. So let's look to the right. So you have G1, S, and G2. This is the interphase part. This is where the DNA is condensing and replicating. Okay. So if you want to know anything about Interphase, there are three parts, G1, S, G2. The most important part is that S phase. That is where cell replication takes place. Okay. If you don't replicate the DNA, I'm sorry, S is where DNA replication takes place. If the DNA does not double, you won't have enough DNA to put in two separate cells. Okay. So interphase makes up G1, S, and G2. S is where the DNA doubles. And then we go through mitosis. Okay. Mitosis is what you already know. It is when you have metaphase, anaphase, telophase, prophase. All right. Those make up mitosis. And then once the cell doubles and creates the new cells, but right before they split, that's called mitosis. And then when they finally split, that process is called cytokinesis. But there's nowhere in this visual where you see the word duplication. And that's because the test is going to make sure that you understand the language, right? So making sure you use the right vocabulary. Could we say in S that it duplicates? Yeah, we could say that. But in terms of proper language, that particular phase is called the S phase of interphase. Okay, does that make sense? All right, just remember, three phases of the cell cycle: interphase, mitosis, cytokinesis. Three phases. And you need to be able to tell what the three phases of interphase are. And that's G1, S, and G2. All right, let's go to the next question. All right, this is a great one. Give me all stages of interphase, or tell me which one of these is not a phase of interphase. What color? Which color is not a phase of interphase? Let me show you that screen again. I forgot I switched. Which of these is not a phase of interphase? That is correct. And I want you to think very simple, right? Interphase. When you think enter anything, it's in between something. So it's the phase in between cells dividing. The phase in between cell dividing, and the phase when they divide is simply mitosis and cytokinesis. So it's just the in between phase. And cells can sit in G1 for a long time until the cell gets big enough and it says, okay, now we need to split in half. Other than that, it will just sit in interphase for a long time and then go into the rest. All right, let's go to the next one. And this is really just a knowledge question. You guys likely went over this in class, and you should be able to just call it from the top of your head. The Galapagos finches have different beaks because of what reason? What color? Blue, teal, as you would say, gold, or red? That's correct. It's all about feeding purposes. So all those birds eat different types of seeds or different types of food within the habitat, which means they need to have a specific type of beak to meet their need for feeding. Very good. And remember, let's go back to this question. All adaptations are all about survival. It's all about survival. So the one in red about fighting, that has nothing to do in essence about surviving. In order to survive, you need food, water, shelter, right? Food, nutrition, some type of shelter, and then most of the time we talk about evolution, which is that change over time to adapt to the changing environment in which you may live or the organism may live. Let's look at this one. Which of these is not a source of genetic diversity or the amount of genetic differences within an organism? That's correct. If you had to describe mitosis as something else, you could simply describe it as cloning. When a cell goes through mitosis, you are producing the exact same cell that you started with. All right, it's a direct replicate. These other options, mutations, the segregation of homologous chromosomes, and independent assortment can all give you a variation, which means that that new product may not be the same as it was before. All right, let's go to some more questions. Let's get out of DNA because we'll tackle that a little bit later. I do like this. Look at this image in the middle. What's happening right here in the middle? And this would lead to a genetic difference. Correct. Crossing over. So those two things would exchange genetic materials, and that would create a brand new organism because it won't have the same DNA as it started with in the parental cell. Let's see. Which one is homozygous recessive? Homozygous recessive. I'm only picking this one because of the language. Just want to make sure you understand the language. Which one is homozygous recessive? That is correct. Two little t's. Homo meaning same, and recessive is always going to be that lowercase letter. All right. Double eyelids are dominant to normal eyelids, which is a little e. What is the genotype for someone who has double eyelids? What would be the genotype or the genetic makeup for an individual who has double eyelids? That's correct. Remember, for each genotype, you have to have two letters, one coming from both parents. If it has at least one dominant trait, is going to appear, and we call that the phenotype dominant. So whether it's two big E's, you're going to have double eyelids, or at least one big E and one little e, it will have a double eyelid. Perfect. You're doing great. All right. Capital T is tall, little t is short. In the Punnett square, um, what belongs in that missing square? What belongs right here in that bottom right square? That is correct. Little t, little t. All right. Looking above and looking to the left. All right, let's see this one. You should be able to do this in your head, but on the test, be okay with on your scratch paper working these Punnett squares out. Implants tall is dominant to short. Perform the following cross. Big T, little t, which is going to be a tall parent to another tall parent. Both are heterozygous. What is the probability of producing a short plant? All right, two tall heterozygous parents cross. What is the possibility of creating a short plant? That is correct, 25%. Now, just for your memory, anytime you have two heterozygotes at cross, the option of getting a short or the recessive trait is going to always be 25%. The option of getting the dominant trait, in this case tall, is always going to be 75%. So 75% tall, or whatever that dominant trait is, 25% short, or whatever that recessive trait is. You don't even have to think about it. If both parents are heterozygous, it's 75 to 25 every time. All right. That's the phenotype, right? But the question here talks about what they look like physically. If we talk about the genetic makeup and I talk about the genotype, do you know what the genetic ratio would be for two heterozygous parents crossing? So it's 3-to-1 phenotype. What would be that genotype ratio? Not quite. So let's look at something. All right, so look to the right. So here we have that same example, right? So when you look at it, you told me that 25% are going to be short, and that's correct because you see two little t's. So that's a 3. So big T, big T, big T, little t, big T, little t. All will be tall. That's 75% or a 3-to-1 ratio phenotypically. If they ask you genotypically, what's that ratio when you have two heterozygous parents? It is 1 to 2 to 1. And now we're talking about not what they physically look like, but what is the genetic makeup. Okay. So you have one that is homozygous dominant, you have two that are heterozygous dominant, and you have one that's heterozygous recessive. Okay. Always going to be the same ratio. So the phenotype ratio for a heterozygous cross is always going to be 3-to-1. The genotypic ratio for a heterozygous cross is always going to be 1 to 2 to 1. Okay? Always going to be 1 to 2 to 1. And I don't care if you change these letters to P's, to R's, to B's, to D's, to X's. It doesn't matter what the letter is. If the parents are heterozygous, that ratio is always going to be the same for both. Okay? Question about that. All right. And simply all I'm saying is I'm just breaking these three that are tall into two categories. One that's tall, heterozygous dominant, and two that are tall, heterozygous recessive. I mean, heterozygous dominant. That's all I'm doing. I'm just breaking that 3 into a 1 to 2 ratio. That's classification. I'm going to skip past that a little bit. I want to get a little bit of ecology, only because you don't have that much classification on the test itself. Let's look at this. This is one of those cell questions. Which organelle is responsible for making using sunlight? You've probably had this type of question since 5th grade. That's correct. The only type of organism that makes its own food through the use of sunlight is or are plants. All plants have chloroplasts and carry out the process photosynthesis. Boom. Okay. If you need sunlight to make food, you must have a chloroplast. Let's see if you know this. There are two main answers to this, but the products of photosynthesis. What do you get from it? And just give me two colors. You don't have to type it all in. What two colors are the product of photosynthesis? All right, she says teal and purple. That's close. All right. If you had to change one of those, which color would you change? And what would you change it to? So which one do you feel confident about? She says teal and yellow. Like, stick with the purple. That's my advice. Okay. Stick with the purple. So what other color besides purple and yellow is correct. Okay, so let's look at this image and think about plants and why we need plants, why plants are so important to people. Okay, so in order to carry out photosynthesis, you have to have sunlight, you have to have carbon dioxide, and you also have to water the plants, right? And when plants go through photosynthesis, it releases or produces, remember, they produce the food which they use in the form of sugar, which is glucose. And then it also releases oxygen that we need as humans to breathe in. Okay. So you need sunlight, you need water, you need carbon dioxide, and it produces the food, which is a 5 carbon sugar or glucose, and it releases oxygen, which we breathe in. And then what do? We breathe in oxygen, we breathe out carbon dioxide. Who do you think uses carbon dioxide? It goes right back to the plants so they can go through the process all over again, right? So we are really very dependent upon one another. All right. So oxygen and sugar or glucose are the products of photosynthesis. All right. This is another easy one. It should be, at least. We just talked about this. Plants need blank to conduct photosynthesis. What do they need? What must they have in the beginning? Correct. They have to have carbon dioxide. We just talked about what they make. We're not going to, ooh, do I want to go through this? I'm only showing you this because they're going to show you this equation. All right. And so you're going to have to almost memorize this equation for respiration. So remember what I just said about humans breathe out something. Look at that. Look at these equations and see if you can see where one of these identify what I just said we breathe out as a proper equation for respiration. Now, so that was great. I like what you did there. So you're looking for these O's, right? Because you remember us talking about oxygen, right? But we breathe in oxygen and we breathe out carbon dioxide. So the opposite of photosynthesis in terms of process is respiration. So where photosynthesis started with water and it started with carbon dioxide, it started with oxygen and it started with sunlight, it's going to be the opposite for respiration. So when you think what humans do to go through respiration, we have to have the pause. I totally said that wrong. You're right. So it needs to start with the O2. And so what you said was yellow. So in the yellow, it starts with carbon dioxide, right? If we breathe in oxygen, you want to look for one of the equations where you start with oxygen, okay? If we start with carbon dioxide, which is where the yellow is, that's really photosynthesis, right? Because it starts with carbon dioxide. It also starts with water. The only thing it's not showing is sunlight. So if it starts with carbon dioxide, that is going to be photosynthesis. Pick another color, but find one where it starts with oxygen and not carbon dioxide. Okay, so let's go ahead and eliminate yellow and let's eliminate red. So let's look at these two where it starts with um, oxygen. So let's break these two down. The oxygen is correct. When you see the C6H12O6, that is your sugar, or that is a carbohydrate that we use to break down to produce energy. Okay. So the difference between these two, just looking at the part before the arrow, is that the blue does not have energy involved. The teal does. The teal says you need oxygen, you need some type of sugar or carbohydrate, and you need energy. All right. We do not need energy. Any organism that carries out respiration does not need energy in the beginning. Okay. However, it will produce energy in the end. Respiration is all about producing a whole lot of energy, because that energy is what allows the organism to carry out a whole bunch of processes. So, like for humans, we need energy to breathe, for our heart to beat, to walk, to move, to battle illnesses because of our immune system. All of that stuff takes place because we consume food. In this example, the C6H12O6, which is a carbohydrate, and then we break that food down and we break that compound apart, it releases energy. Okay. So you're going to start with oxygen. You also need some type of carbohydrate. And at the end, you're going to produce carbon dioxide, which we breathe out. It goes back to the plants. We release water, right? We sweat a lot, right? But we also produce water in the process of respiration. And then we're going to produce a whole bunch of energy. Let's test. See if you can tell me this. What is the form of energy that you produce in respiration? All right. What is the form of energy you produce in respiration? It's 3 letters. Blank, blank, blank. When you think of cellular energy, what 3 letters make up cellular energy? I see we got another person who's watching on YouTube. So welcome. We're just kind of doing some basic reviewing right now before we get into some of those really, really tough questions. So anybody can answer. What are the 3 letters of energy in the cell that you produce in respiration? And I want you to think ATP. Okay. Anytime you see ATP, ATP is a form of energy that you produce or create in respiration. So I could replace the energy in the equation where the blue is, and I can put ATP. So I could say you start with oxygen and a carbohydrate, and then you yield, which is what the arrow represents, yield or produce carbon dioxide, water, and ATP, and it would still be correct. Okay. Question about that. And I can, if you take that blue box, that equation, if you flip it and add energy to the beginning, so if you put CO2, water, energy, and sunlight, and it will produce oxygen and a sugar, which is at C6H12O6. That is photosynthesis. The equations are exactly the same. They're just flipped. That's it. The only thing you need to add is sunlight to the beginning of the equation for photosynthesis. Other than that, they have the same molecules in there. They're just on different sides of the arrow. Okay. All right. So based off what we just said, I don't want you to think just about humans. What do you think is the main goal of respiration? Blue, teal, gold, or red? What's the main goal? Exactly. The main goal is to produce energy. All right. I think we've beat that one. Let's move on to some ecology questions. So give me just a second. We're going to flip. We're going to pull in some ecology. And let's see what we got here. Let's see if you can do this one. And you can put the numbers in the correct order in the chat. All right, put these in the correct order from the smallest to the largest. Well, I guess you would do colors. So I do colors because the numbers are telling you the order. So what color would you put first? Let's see if you would have something different. Look at it carefully from the smallest to the largest. All right, so it's purple, blue, no, so blue, purple, yellow, teal, red. Now, was this one difficult for you? Before I give you the answer, did you find this difficult? All right. One of the reasons you found it difficult probably is because you overthought the question or you overthought the ordering because it's already in the right order. Okay. So you don't have to move anything. The smallest part of an ecosystem is an individual organism. When you get multiple of the same organism, then it goes from an organism to a population, right? So if I have one dog species, if I have a group of the same dog species, that is a population of dogs. When I add in other dog species and some other things within that area, it makes up a community. So I'm getting bigger and bigger. From community, I go into an ecosystem, and then finally the biosphere, which is basically the earth itself. All right. So think of ecosystem, think like United States of America. And then I could add in biome. The biome would be North America. The whole continent would be the biome. And then biosphere takes it to the next level. Okay. So don't always think you have to move things around. Look at it. If it makes sense to you and you feel like it's right, go with it. I always tell people, trust your gut. Trust your gut. If you looked at this one and you felt like, man, I think this is right, more than likely it is. Trust yourself. There's something in your brain that I always say connects to the answer. You may not always be able to explain it, but trust yourself. I don't want to do, let's see. Because these are just definitions. And I don't want to do definitions. Let's do this one. Now, I will tell you, this one is not in the correct order, okay? So I want you to think the cycling of matter and energy throughout an area, okay? The cycling of energy and matter throughout an area. Where would this cycle start and where would this cycle end? So give me the colors. This is not in the correct order. Where would you start? All right, red, that's correct. Always start with the source of energy for everything. That's correct. So the sun gives energy to the plants to carry out photosynthesis, right? Then the plants are eaten by insects. The insects are eaten by small or large animals. And then when those organisms die, they are broken down and reconsumed by the earth through or by decomposers. Boom. You got that one. Perfect. Perfect, perfect, perfect. Um, let's look at this. Let's see if I can find a better image for that. I want to talk about the energy pyramid, but I can do it from this actual diagram here. All right. So looking at these, I want you to tell me where these organisms fall um, in the correct order. So if we are looking at the grass, does the grass consume anything? Does the grass consume anything? No. So at the beginning of any cycle or any type of food chain or food web, you're going to always start with the producer, which is always going to be a plant first. Sometimes they start with the sun, but most of the time they're going to start with the actual producer, right? So in this case, the grass is your primary producer. How would you describe the mouse? And based on the image, the arrow is telling you that the mouse feeds on the grass. Therefore, you're correct. It is the primary or the first consumer within this food chain. How would you describe the snake? If the mouse is the primary consumer, that's correct. The snake is the secondary consumer. Therefore, the hawk would be the third or the tertiary consumer. Absolutely correct. Now, if I'm looking at the same food chain, this is a conversation. They can use the same diagram to ask you multiple questions. Okay. The grass makes its own food because it goes through photosynthesis. Therefore, the amount of energy housed at that level is 100%. Okay. 100%. Through each food level, only 10% of energy is transferred. So if the grass produces 100%, what percent of energy does the mouse get from eating on the grass? Then you just give me a number. If 10% is transferred through each level, 10% of energy, if the grass starts with 100, how much does the mouse get? Correct, 10. Okay. And remember, the grass is the source. So the grass has 100, the mouse gets 10%. How much would the snake get? 1. And how much would the hawk get? There we go. Now, if there was another one that ate the hawk, it would get zero point 0 and it would just keep going, right? Very good. Very good. All right. Now, you may get the same questions, not from a food chain, but you may get it from the same food or from a food web. Just be able to identify several food chains within the image that they give you. If it's a food web, you're going to do the same thing. Okay. You should be able to give the food chain, talk about what is a primary, secondary, tertiary consumer. Also, be able to talk about the amount of energy that's transferred through each of those food chains. Now, the other thing you may talk about is the biomass pyramid or also the pyramid of numbers. Okay. You just need to know the greatest amount of mass is always going to be at the producer level. Okay. And the greatest amount of, in terms of number of organisms, is always going to be at that producer level. As you go up the food chain, the numbers of organisms decrease and the actual mass increases too as you go up. Okay. So just know I could have 10 million, 1 million, 200,000, 8,000, and 20, right? Those numbers just need to go up as you go up the food chain. And then the amount of mass. Mass is typically in kilograms or grams, but whatever it is, it should decrease as you go up. Now, this is a great example of a predator-prey graph. You should be familiar with these. You should have seen these in class. These are very common, and you're going to likely see it in the test. Okay, so this talks about the relationship between the predator and the prey. So looking at the graph to the left, and you will have questions where you would have to go in and move these actual words into the actual sentence so it can make sense. So it says the number of blank affects the number of blank. So very simply, this should be the number of predators, which is in blue, impacts or affects the number of prey, which we have here in yellow. Okay. As the prey numbers blank, so does the number of predators. As prey numbers blank, so does the number of predators. And it's really self-explanatory. If the prey increases, then the number of prey increases. Why? Because there's more available food. So when predators go through reproduction, they produce more babies, but those babies also have food to eat because you have a whole lot of animals to eat on. But if the prey number decreases, the amount of available food in the area is going to drop, which means some of the predators are going to die because of starvation and they're competing for the limited amount of food. You should be able to take this same scenario and apply it to this. So when this yellow bar is going up, what do you see? The blue bar is going up. But when that yellow bar drops, what happens to the prey? It drops. All right. So they have a direct relationship. When you have a lot of prey, you have an increase in predator. When you have a decrease in prey, you're going to have a decrease in predator. Okay. They go hand in hand. All right. Questions about anything so far that we've covered? We've hit maybe about 20 questions. Any of these questions you want me to go over or maybe talk about a little bit more? All right. So now let's go into some more difficult questions, okay? So take a quick look again at the domains that you're going to be tested over. And let's hit the first question, okay? So this is covering standard 1B. It says you're going to develop and use models to explain the role of cellular production, which we just talked about. Remember, we talked about that mitosis in maintaining genetic continuity. So look at the image, okay? Just look at the image first before we even get into the question. What do you see inside this circle? And number one, what's inside of that circle? That's correct. All right. That's a great start. Now, look through the numbers 1, 2, 3, 4, 5, and then you end with 6 and 7. Okay. So here's a question. Which statement correctly uses the model to explain how mitosis maintains genetic continuity? And I'll blow this up a little bit for you. How does the image or the model explain mitosis maintaining genetic continuity? Continuity mean that what I started with, I'm able to see again at the end of the process. How does mitosis do that? A, B, C, or D? Let me make that smaller again, so just in case you need to see the image, you can. All right, you say B. So let's look at B. So the key word when I look at this in B is the word crossing over. Okay. When you have crossing over occurs in meiosis. Okay. Meiosis is reproductive cellular division. And meiosis is important because you have variability in your offspring, which is why you and your siblings don't look exactly alike. Because more than likely, during the process of meiosis, those cells crossed over. Okay. Remember, in mitosis is all about, what did I say? Cloning, producing the same cell over again, including the genetic material in that cell. Okay. So when you look at B, when you say crossing over and you see the word diversity, you don't want to see that in mitosis. You don't want any, or will not have any, diversity in mitosis. So when you saw those two words, you should have been like, not the answer. Okay. C says when the nuclear membrane reforms and each nucleus becomes diploid in number. Well, if you look at it, it's already diploid. Okay. So we don't have to talk about the diploid amount here. Because the diploid number is, in terms of it replicates where? When you get to number 2. So by the time I get to 2, not 5, I now have 2 of each DNA strand. Okay. So that's incorrect. Letter D says the independent assortment that is represented in 2, well, they're not even separating yet. So that can't happen. The independent assortment starts happening once we get to 4, right? They begin to separate and assort along the ends or the poles of the cell. So that's not correct. But the answer is A. The chromosomes in cell 1 are identical to what I see at the end in the brand new cells at the end, which are 6 and cell 7. Okay. What you start with, you need to end with in mitosis. They should look exactly the same. If they don't look the same, then you know the question is not related to mitosis. It's related to meiosis or meiosis, however you want to say it. Okay. If they're different, then it's meiosis. If they're the same, it's mitosis. Okay. You will have some graphs, a lot of graphs on the exam. They'll try to give you a lot of visuals, where it's graphs, pictures, tables, or something of the sort. Maybe even some Venn diagrams, right? Some type of graphic organizer to kind of help you filter in this information. I'm not going to do this one just because it's a longer question, and I want to use our time. We're already over time. So let's look at. Let's see if we can find a shorter one. But the ones on the test are going to look kind of like this. Let's look at this one. Number 8. Students studied Mendel's laws of segregation and independent assortment. They created a Punnett square to show a cross between homozygous furred mouse, all right, capital F, and a homozygous furless mouse, little f, little f. The students then created a second Punnett square for a cross between 2 heterozygous F1 mice. Okay, so Here's the first cross, which is the homozygous fur and the homozygous furless. And then in F2, which is the second cross, they cross two heterozygous organisms or two heterozygous mice. Which question could be answered using the information from the Punnett square? And the question simply asks, what question can you ask from the Punnett squares? Don't infer anything extra. Answer the question. Correct. Some of these other questions you can't answer from the Punnett square because they don't give you that kind of information. But based on the Punnett square, I can ask, can a furred mouse produce a furless mouse? And in this case, the answer is yes, because in F2, both of these are heterozygous, but they're both furred and they can produce a furless mouse. And what percentage can they likely produce a furless mouse? What is the percentage that they can likely produce a furless mouse? There we go. Boom, 25%. Exactly right. Let's look at this one. So it says the model shows part of a process that uses transfer RNA. So which description explains the role of tRNA in the process shown in this model? I will tell you this model relates to transcription and translation. Okay? Particularly, this is translation. What is the role of the tRNA in translation? This is a little bit harder because it's assuming you know these processes already. All right, so you said the tRNA scans the developing peptide to make sure that the sequence of the amino acids matches the messenger RNA. That would be correct for a particular type of RNA, but not the tRNA. Okay, so let's simply talk about this. So you got two processes, transcription and translation. In transcription, you're going to take a single-stranded DNA and you're going to transcribe it into a messenger RNA strand. So one single messenger RNA strand. Remember, in RNA, those we have no more thymines, but we have uracils, right? So we have A, U, C, and G in that messenger RNA strand. That messenger or that mRNA is then translated into a protein, right? So DNA to RNA is transcription, RNA to a protein is translation. What is a protein? It's several amino acids put together, okay? Now, when you create those proteins, you need the tRNA to come in with the amino acids. So here's a tRNA. These circles represents these amino acids. Okay. So the correct answer is A. The tRNA delivers the amino acid to the ribosome. And then those amino acids connected make up a peptide or make up the final protein. All right. So it just carries in that amino acid on the tRNA. It lands on the ribosome. And once the amino acids connect, the transfer RNA just goes away and it goes to get another amino acid. Okay. So the tRNA carries the amino acid in translation. All right, not in transcription, but in translation. That was a little bit tough, right? Takes a little bit more thinking, but just now that you know that, make sure you review the transcription and the translation process. But I hope it made sense. Let's see if I can find you a quick visual between the two. It may not go through all the steps of the two, but at least you can visually see what you're producing. Let's use this one. All right, so look to the left. If I can make this, no, we'll keep it there. So you will look here at the very top. I'm going to take just one part of the DNA, right? And I'm going to transcribe it through transcription into a messenger RNA strand. That messenger RNA strand is then translated into a polypeptide, which in common language is the actual protein. But in order to do that, you have to have your tRNA and your messenger RNA. So I want to find something that talks about the tRNA because it's such a particular process. You got to be specific when you're looking for it. So most times you'll see something like this. So when you see that you have these amino acids, you just have to know that these amino acids were carried by a transfer RNA. This is a better picture, okay? So this doesn't look like the little block. They could look multiple ways, but when you see an amino acid being carried by something, you know that something is the actual tRNA. And then we had already talked about codons, right? So 3 bases, so AUG. In that tRNA, the tRNA will have a complementary codon to what's in that strand of RNA. It has to pair up perfectly. If it doesn't, it can't drop off the amino acid. So this is AUG here. Let's make this bigger. Let's look here. Look at this one, okay? So here you see AUG, right? AUG. The tRNA has to have a base pairing that's exactly the same or that complements it. So A pairs with U pairs with A, and G pairs with C. That lets the tRNA know this is exactly where I need to drop off this amino acid. It will land right there on that messenger RNA, and then you'll have another transfer RNA that will come in and bring in another amino acid. And then those amino acids will join, and then the tRNA just drops off. It disconnects and goes away. Okay. And what you see where it says this small unit, imagine that the messenger RNA, which is this with all the bases, is like a train track. And the small subunit is the train. The small subunit will just go down from 5 to 3 and start bringing in transfer RNAs until it gets to the end. And then you should have the product should be what we call this polypeptide, which is just a bunch of amino acids connected. But they only connect because the tRNA comes in and brings in those amino acids one by one by one until it's done. All right. If you want to get even more technical, the small subunit is called a ribosome. Specifically, it's a type of RNA. It is called the rRNA. All right. So you may get a question like, what are the 3 types of RNA that are involved in translation? Well, you can see it right here: tRNA, mRNA, and rRNA. Okay, T, R, and M are all involved in translation, and that is the process of making proteins. Does that make sense? I covered like 2 weeks of like classroom study in like 3 minutes, but it should all be coming back to you, right? It's about really understanding the steps of each of these processes, and it has to make sense that it's so fluid that if you see it, you should be able to expel or draw out some of the information to answer the question correctly. That is my goal. All right, let's do one more and I'm gonna let you go. It's already 8:50. I don't want to wear your brain down. Let's see. Let's see. Let's look at this one. It's going to be another one. It's kind of hard, but we're going to walk through it together. All right. So this is number 12. The diagram represents a model of how bacteria become resistant to an antibiotic, right? Remember, bacteria are bad for your body most of the time. And so if the bacteria is resistant to an antibiotic, which means the antibiotic is not killing it, which means you're not getting better. All right. And so it says the bacteria is surviving the treatment, which best explains how the indicated step in the model allows bacteria to develop resistance. So they're looking at the one that has a square around it. How does this particular example indicate the step of how the bacteria develop resistance? How do they become resistant more than likely? And I know there's a lot of heavy language in there, but think about what I talked about earlier. So the alleles for antibiotic resistance become dominant over recessive alleles. That may be true, but the key word in the question or in the answer you said is that the alleles, they become dominant. Well, what would cause them to become dominant over recessive, right? What would cause that type of change within the bacteria where they will become resistant? So give me another choice. What would cause something to change? There we go. Exactly correct. A mutation creates change. So somehow within that DNA of the bacteria, there was a mutation that occurred. Because of that mutation, the antibiotic could not attack the DNA the way it normally did because it was not the same DNA. It had changed. And then what I had left after the treatment was two cells or two bacteria cells that survived. And what did they do? They went through replication and they began to reproduce. And so now I have a bunch of brand new bacteria cells that are all resistant to the treatment. Good job. Good job. Good job. That's how I want you to think about it. Something changed. So you got to think about what would cause a change in something. It would be a mutation. All right. Any questions? We're going to go ahead and wrap up. Um, you've got about a month. Would you like to do this again before your test? I hope this was helpful. All right. Um, I don't know how you found me. I don't know if you found a flyer, if your mom found it, or a teacher told you about it, but let them know I'm going to do this again before the test, probably in about 2 weeks. So I'll drop a flyer again. It'll give you the date and the time. The link may be different, so just make sure you check the flyer for the proper link. Okay, but we'll use the same format. Good. All right, awesome. Have a great school week coming up. Get some rest tonight. Be ready to go. I want you to go to school and work hard every day. Don't take a day off because this test is fast approaching and I want you to do really well. Okay. Dr. Holmes is here for you. I really appreciate you for joining in. Tell all your friends next time to come back and join me so we can have more people in. All right. Thank you. Have a good night. Okay.